I hand over the session to the session chair. We can't share.
This has been a very important and very interesting session which we have today. This is also the part of the seminar talks, very of the researchers also presenting the best in a short time. So what we talk about it, and this is a mixed bag of everything together. And I hope you really enjoy the session and if you have questions at the end. The first speaker is Lavini Roy. And we'll be talking on underwater soundscapes, caustic response to thunder and conflict in the environment. I request Ashish to currently working as a senior project fellow in the project. She is also a PhD scholar from Boston Institute. Just one announcement, we have still time. We have 5 minutes.
Good morning, everyone. I am Gargi Roy Chaudhary, and today I'll be presenting about underwater soundscape and acoustic response of Ganges dolphin in noisy environment. Ganges dolphin are present in the Ganga, Brahmaputra, Karnafuli, and Sangu river system. They are functionally blind and can detect only light and dark, and they heavily rely on echolocation to detect threat and for prey. They constantly emit echolocation sounds, and they are endangered species and also scheduled species under the Wildlife Protection Act. They face many threats, but today we'll be talking about its response to noise. So for this, our study area was River Hooghly, which is a human-dominated riverscape. And since colonial times, Kolkata port paved way for the import and export of goods. Dwarkanath Tagore, also known as the grandfather of Rabindranath Tagore, was a pioneer in establishing shipping companies with the British traders for import and export of goods. Waterways is considered as a cheaper mode of transportation, and so this dolphin habitat is now a part of National Waterways 1. Due to increase in the navigational facilities, we see that there is 16% increase in cargo ship movement in the National Waterways 1. And since the mode of transportation is cheaper, we see that throughout the river Hooghly, there are ferries of different capacities. So how did we record the traffic and related it with the dolphin behavior? So for traffic recording, we visually observed the boats, and in absence of the researcher, camera trap observed the boats. And there were F-pods, which are passive acoustic monitoring devices. They were anchored to the river bed, and they recorded the dolphin clicks. And then we did an overlap plot to see the temporal activity of Ganges dolphin with boat traffic. And we see that small boats are used for fishing and sand mining, and they are active during late night and early morning periods. Medium-sized boats are ferry boats, which are active during peak office hours, and large barges that carry coal and fly ash to Bangladesh also show periodicity in their movement. And dolphins tend to avoid this high boat activity period, and they shift their peak to avoid the boat movement. We also recorded the soundscape of the river. For that, we used hydrophone, and the clicks from the hydrophone was matched with the exact clicks from the F-pod that records the acoustic parameters of river dolphins. And we found that with the increase in sound level of the river, there was a positive correlation with minimum frequency and interclick interval of the dolphin echolocation clicks. So whenever a boat passes, we see that the sound pressure level of the river rises. When it is an ambient, ambient means when there is no boat present, the sound pressure level of the river is 139 decibels. But whenever a boat passes, it increases up to 190 decibels, which is approximately 50 decibels rise due to the motorized boat movement. We did generalized linear model to account for frequency change whenever there is a boat movement and also interclick interval. We found that there was 10 kilohertz increase in the frequency range of dolphins and 1000 microsecond, which is 0.001 seconds, increase in the interclick interval. Though it seems a very minor change, but it affects the dolphin and can cause immense stress to it. We also recorded dolphin acoustic parameters before, during, and after the boat movement. And we can see that when the boat is passing, the frequency and the interclick interval is rising and then falling. So in conclusion, the dolphins uses higher frequency in noisy water because most of the environmental noise is in the lower frequency, and thus to avoid masking, it has to communicate at a higher frequency level. Although there are checks in place like reduction in speed of the boats and different design of the boat in order to counter with the sound produced by the large boats, still there is a chance that it will have the same fate as the extinct youngster dolphin, which the major cause of its extinction was increase in shipping. Thank you everyone.
Next speaker now we have Ashi AS, and he will present on can data machine learning approach aggrandize the connectivity analysis and everything of tiger connectivity in digital
Thank you for the introduction. The topic that I am going to discuss here is, can data-driven machine learning approach aggrandize the connectivity analysis, an avenue to tiger connectivity in eastern Vidarbha landscape? Coming to the introduction, as we know about biodiversity conservation, it requires an effective management of wildlife population and understanding of complex relationship between wildlife and ecological components. These ecological components are the major drivers for the biodiversity conservation. Incorporating data analytical approach like artificial intelligence, machine learning, or deep learning into ecological data sets helps in understanding the hidden insights from the data as well as fine-scale information from the data. With this as a background, we tried to incorporate machine learning algorithm for performing the connectivity analysis of tiger in eastern Vidarbha landscape. Thus, our aim is to examine whether the connectivity current map prepared using this approach is aggrandizing the connectivity analysis or not. The term aggrandize means augment or enhance. This is our study area, eastern Vidarbha landscape, comprising of districts of eastern part of Maharashtra. Coming to the methodology, We used the datasets from anthropogenic, bioclimatic, and habitat domain to perform the circuitscape connectivity analysis. Circuitscape is the software used by the researchers working in the domain of connectivity and conservation. It takes habitat raster and focal nodes as an input. It uses electric circuit theory algorithm to generate the connectivity current map. The connectivity map thus represents the least resistance path used by the wildlife species to move through the fragmented habitats. Researchers are using various approaches for generating the habitat raster, that is the input to the circuitscape model. Researchers are using GIS-based approach, analytical approach by assigning weightage to the geospatial parameter. Researchers are also using species distribution model output of Maxent. In this study, we used machine learning algorithm for performing the connectivity analysis. We use 4 machine learning algorithm, random forest, Navi Bayes, extreme gradient boosting, and K nearest neighbor for generating the probability prediction map. That is our habitat raster. Coming to the methodology workflow, we have input geospatial parameter. We have camera trap presence and absence data as the target. We implemented the machine learning algorithm and we've done cross-validation using cross-validation data sets, and we choose the best appropriate model. The best model is the Navi-based model, which is having an accuracy of 92.6, which surpasses all other models. The Navi-based model output we used as an habitat raster. The Navi-based model output we used as an habitat raster. That is our probability prediction map, which we are giving as an input to the circuitscape model to get the connectivity current map. The connectivity current map obtained for the eastern Vidarbha landscape is shown in this slide. Here, red represents the region of high connectivity and blue represents the region of low connectivity. We have color gradients from 0 to 1, color gradients representing the values from 0 to 1. As we observe closely through this connectivity map, we can see more fine-scale details are able to capture from this connectivity map. Also, the corridors can be mapped more precisely. We make a comparison with the already available connectivity map generated by Mondal et al. In the year 2016. They used the species distribution model output of Maxent and circuitscape model to generate this. While comparing, we can see more fine-scale detail is available in the machine learning-based output that helps in identifying the mitigation areas in the village level. Also, the corridors can be mapped more precisely, which helps in performing the conservation activities more accurately. For further comparative analysis, we classified the connectivity area into different classes ranging from low to very high. And we found the area, and that is represented in the graph here. And we found that while comparison, there is a gain of 39% of very high connectivity area. Also, a loss of 11% in low connectivity area. For further validation, we overlaid the radio telemetric data into our connectivity map, and we can see the movement of tiger through this fine scale map. Also, we can see the tiger is moving, and the white region represents the low connectivity area, and it is not going to that low connectivity area, and it is passing through the fine scale high connectivity area. Coming to the conclusion, what makes our study distinct? It is because of the capability of the machine learning model that is generated from scratch to generalize the input information. Also, the machine learning model is able to capture fine scale information, and we have the option to do fine tuning of the model, which means using random or grid searching, we are choosing the best appropriate model parameter to get the high accurate model. This is called hyperparametric optimization. Also, this model is having the capability to capture complex non-linear relationship from the input data. In the way ahead, we have a plan to extrapolate our work throughout the entire Central India. Also, we will use telemetric data to perform this machine learning analysis. Also, we have 4 machine learning model output. We will do some ensembling to capture more accurate results. I would like to acknowledge the director, dean, research coordinator, and the following people. Thank you.
sir. Hello everyone and good morning. Today my topic of presentation is estimation of density ungulates in a tropical semi-evergreen forest, which is a case study in Pakke Tiger Reserve. So there are many methods available for population estimation of ungulates like sign survey, line transects, and also camera trapping. While line transect is very much viable in open forest and accessible areas, there are some challenges when the landscape has dense vegetation and low animal encounters. So keeping this in mind, the objective of the study was to provide density estimate in a robust way using a method which is appropriate for the study area and which will also reduce the human effort. So the study area, Pakke Tiger Reserve has three ranges which are Sejosa, TP and Rilo, and our sampling was mostly done in Sejosa and TP range and a portion of the Rilo range. Coming to methodology, the study area was divided in 1 square kilometer grids and cameras were deployed at the centroid based on the accessibility of the point, and placement of camera was random to animal movement, and 79 traps were used, accumulating almost 8,000 ungulate images were captured, which is 75% of the total image. And camera calibration is used to digitize the view in front of the camera to later tally it with animal images, which is used, a pole and a measuring tape is used for the digitization purpose in front of the camera. And animal distance measurement, during animal distance measurement, the resultant reference image is tallied with the animal image to get accurate distance measurement using image and animator software. And this animal distance is later used in distance software to model and get the effective detection radius, which is later used for density estimation. Coming to results, sambar deer was captured most, almost 4,500 captures of sambar was there, but hog deer, sero and goral capture was so less that density estimation for these three species was not possible, and effective detection radius was highest for wild pig and lowest for the barking deer. Coming to density estimation results, as I was showing, sambar had the highest density at the tiger reserve and wild pig at the lowest density. And interestingly, while comparing the relative abundance index, which is capture per 100 trap days with AITE exercise, All India Tiger Estimate exercise of 2018, we saw a significant increase in capture per 100 days for all the ungulates, and notably Himalayan Goral was also captured, which was not previously captured. And we used wild pig as a model animal to compare the density estimate using camera trap in two different methods, which is random encounter model and camera trap distance sampling. And you can see there is no significant difference, as the upper and lower limit in 95% confidence interval is almost overlapping. While comparing with the line transect study, there was no much difference in precision. It was almost similar, but the area coverage of CTDS camera trap distance sampling was a bit more. Coming to the conclusions of my studies, we can say that CTDS is a powerful tool because it reduces human effort, hence it gets rid of the human error bias, and compared to other conventional methods, it also covers a larger area. And for the forest patches where there is dense understory and impaired visibility due to vegetation, it will be a very useful tool. But ruggedness of the terrain and inaccessibility needs to be mitigated. So to infer about CTDS's effectiveness on a finer scale, further studies is required. So I would like to acknowledge the following people.
And the next presenter is Dehradun. He's going to talk about the integrated taxonomy report in the grazing activities covered on
Thank you, sir. Good morning, everyone. Understanding and identifying the diversity may act as an insurance against nameless extinction. However, overcoming this knowledge gap is challenging when it comes to a group with high level of cryptic diversity. Thus, our study aimed to delineate the species diversity of Northeast India using multiple line of evidence, that is integrative taxonomic approach. Northeast India often considered as a biogeographic gateway between two mainland, that is Southeast Asia and India, and hence this region is a region of reticulated evolution, and this region lies at the junction between two globally recognized biodiversity hotspot. However, the role of biogeographic features never have been tested phylogenetically, especially for herpetofauna. So, to address our questions, what is the actual diversity present in this region, what is the distribution pattern, and what is the role of biogeographic features of this region. We conducted our study during 2019 and 2023, where we surveyed at least 40 localities, which includes 20 protected areas. In field, we used nocturnal visual encounter survey, around 200 visual encounter surveys, and surveying manually for the species in different kind of habitats such as stream habitats, forest trails, etc. To delineate the species, we used multiple line of evidences involving morphology, osteology, behavioral tool, and molecular tool. For morphology, we studied the collected specimens under microscope, where we used at least 40 meristic as well as metric characters. And for osteological characters, we prepared the specimens using clear and staining method. As a behavioral tool for cryptic environment species, we used real-time bioacoustics, which were recorded in field with a recorder and unidirectional microphone, which were later analyzed and visualized in Raven Pro software. For molecular tool, we used maximum likelihood and Bayesian approach involving mitochondrial as well as nuclear genes. So far, we have recorded 300 species from this region. Also, we report a significant increase in diversity in the protected areas, and for some protected areas, at least 10 protected areas. This is the first systematic survey. Our study leads us to describe 8 new species of herpetofauna from this region, including 3 amphibian species and 5 reptile species, which includes 1 new genus. Also, we rediscovered two terrestrial snakes from the Assam Valley after more than 100 years. Our preliminary biogeographic analysis shows that for the arboreal taxa such as these cat snakes, the Brahmaputra Valley act as a filter, where these sister lineages are separated by this Brahmaputra Valley. Similarly, we found same result for agamid lizards, which were separated by this Brahmaputra Valley. And for Raorcystis, which is a direct developing bush frogs, these valleys separate these species into 2 clades, which shows latitudinal dispersal rather than longitudinal. And there is a meeting point of these 2 clades in the far at the eastern part of this valley. But in case of natricine snakes, this valley may not act as a dispersal barrier, which shows by the shallow divergence among the populations distributed across this valley. And for the Southeast Asian groups like the fang frog and rain snakes, this valley act as a strong barrier and northwestern distribution limit. Coming to the conclusion, our study addresses Darwinian shortfall, Wallacean shortfall for many unknown taxa, and which describe many new species to science, and also shows the role of Brahmaputra River acting as a barrier and as a filter. Also reported first systematic survey in Northeast India, especially for the protected areas. However, our study envisages further systematic survey to better understanding the role of biodiversity features and to know the actual diversity. With this, I would like to acknowledge the following people. Thank you.
We have Mrs. Dr. Sunil Hassan here, who is presenting on assessing dolphin quality and requirement. Dr. Sunil really completed her doctoral thesis on the quality of undergraduate, and she is currently working as project associate in her project.
Thank you for the introduction. Good morning, everyone. Yeah. So following my colleague's presentation on how Ganges dolphin is affected by the habitat alteration. I will be talking about the direct killing of Ganges dolphin, which is another threat to this species and is a major concern for the conservation of the species. So coming to the question, why the dolphins have been killed? So it's basically killed for one particular reason, is that the dolphin oil has been extracted from the blubber of the animal, and it is used as a bait to lure a particular kind of catfish. And this has been reported in various instances from all over the country. And also we got some snapshots from the social media. So Bin community fishermen, so these are the major fisher communities which are actually involved in such kind of practices, and they belong to Bihar state so far as known to us. And they have also migrated to parts of Assam and Bengal. And in a study from 1999, it has been recorded that about 500 fisher folks from Assam were identified, which are actually associated with dolphin oil bait fishing. And in that particular report is also been mentioned that about 20 to 25 liters of oil can be extracted from a moderate size dolphin. So bringing all those inferences, scientific inquiry was done to assess the dolphin oil bait fishing in Assam. A recent study has also confirmed that there is a prevalence of dolphin oil bait fishing practices in Brahmaputra river, and the target fish species were Clupozoma gorva and Etrophicus vacha, which are also known as Neria and vacha respectively. Dhubri district of Assam is the major bin, we can also call this the hotspot of bin fishermen community. So we did a snowball sampling in this particular district, and to identify the fisher folks which are associated with dolphin oil bait fishing and also their fishing grounds. We did a semi-structured questionnaire surveys to understand the socio-economic condition of these fishers and the fishing trends. We have identified 146 actual fisher folks which are involved in this kind of fishing activities, and 99 of the fishermen have agreed for our interview. The major result of our study has revealed that there is an extensive use of dolphin oil bait in Assam throughout the entire Brahmaputra valley, starting from Dhubri to Tinsukia district of Assam. And we take into consideration some other information like fishing days in a month and year, and average oil bait, oil extracted from a dolphin. We came to some conclusions like there is a requirement of at least 1,164 liters of dolphin oils each year to sustain these 146 oil bait fishermen. And also when we convert this number, this liters of oil into number of dolphins, we have found that 46 numbers of dolphins have to be killed each year to suffice the requirement. So coming to the death statistics, which reveal that in a that record data of about 14 years, we have found that net entanglement and poaching are the two major contributors for mortalities in Assam, and 61% of net entanglement is responsible for the mortalities, and 16% is for poaching. And one interesting thing that we note here is the 61% of net entanglement, which the causes of mortality, is also can be considered, which are end up into the dolphin oil extraction process. We have estimated 1.3% of decline in population of dolphin each year from the Assam dolphin population. There has been practiced alternative oil bait which is tested and got significant results for in field. However, the pressure on the targeted fish species is continued because of this practice, and hence it is not a recommended recommended solution for the conservation of dolphin. So the socio-economic conditions of these fishermen is very poor, and with an average annual income of less than 1 lakh, this oil fishermen, actually all the fishermen of Assam have to pay about 25% of their daily income to the concerned leaseholders, and they also have to pay 25% of their income, daily income to boat rent and other relevant expenses. So they are left with only 50% of their daily income. And this Neria fish has a very big fish demand value in Assam, and for which these fishermen were also very much dependent in Neria fishing with dolphin oil bait. So this Neria fishing has an annual turnover of 3.16 crores in Assam. So coming to the recommendation that has been made by this study is alternate oil is not the solution, and there should be a complete ban of oil with fishing to conserve the species. And for this, we need a vigilance system to monitor the use of dolphin oil. So obviously there will be effect on the socio-economic of the fishers. So an adequate alternative or livelihood and through skill development programs would be given to these fishermen. And of course, we have to ensure so that this particular fisherman receives all the benefits from the government welfare schemes for the sake of the conservation of the species. With this, I would like to acknowledge these following people. Thank you.
Knowledge who's already on senior status, any pattern factory of the natural property, which is not that, and was that landscape.
Good morning. So these immortal lines from Kaviguru Tagore actually implore us to understand our immediate natural surroundings even more and appreciate it. And on that note, I would like to talk about the simian amidst the sapiens, a species in our very own backyard, which evokes a lot of mixed reactions amongst us. So the rhesus macaque, an old world monkey, last shared a common ancestor with us humans some 25 million years ago. But with the largest natural geographic range for the species, they have seamlessly become interspersed in human-dominated areas. So in 1989, they, along with the long-tailed bonnet and the toque macaque, were classified as the weed macaques, which allowed them to access anthropogenic foods and become synanthropes to live amongst us. India has observed spurts in macaque populations in northern India, and it also faces significant financial losses due to human-wildlife conflicts. But there needs to be serious considerations regarding the present management practices for the species. So we asked, how do macaques actually utilize a human-dominated landscape? My study area was the 16 kilometer square around the Wildlife Institute of India campus, which comprised of a mosaic landscape where we have been studying two socially distinct groups since 2018. We collared two adult females from our study troops with GPS GSM loggers, which were taking fixes at 15-minute intervals, and we obtained data for at least 2 months. And then we find that the troop B actually shows a higher home range and core range in comparison to troop A, with significant overlaps between the two troops. But interestingly, troop B had a higher percentage of forested areas within its range. The daily travel distance, which is the total path length, showed a significant difference between the two individuals, but no difference between seasons. And the daily displacement showed no difference across individuals as well as seasons, which goes to show that even when they are traveling around 2 to 2.5 kilometers a day, their displacement is very small, only around 300 to 325 meters. So we calculated the habitat selection that they were showing as per 5 land use land cover classes in this particular area, and we find that the anthropogenically modified habitats of built up areas and especially the roadsides were being used more than it is available. Site fidelity is very important for this species because of the sparse distribution of refuge sites as well as places where they can actually access food resources. And we found that this graph shows that there is a concentration of these particular areas in the core range, especially across a 50-meter radius, where Troop B, which is the forested troop, used around our root site around 5 times and A around 6 times. So this led us to ask the question as to why and how they were actually moving and what strategy they were showing in utilizing the whole place. So the first is the Brownian movement, where there are similar size step lengths and probably the resources are more evenly distributed. But then there's the non-Brownian movement, where there are long step lengths interspersing the shorter ones, and this could lead to a patchy distribution of resources which they try to utilize. So this is the map as to how they have been traversing in my study area, and we find there are certain spaces which show a higher concentration of trajectories, while certain spaces are being used only sporadically. So again, the mean step lengths were somewhere around 50 to 60 meters, but even then there was a high concentration of step lengths which were at least 4 times to 8 times of the means. So this led us to analyze the step lengths and find that whether they were showing a long tail distribution or a exponential distribution, which is Brownian movement. So we find that even though they are traversing in a very small region, they are showing long tail distributions, basically the power law and the log normal, based on the comparisons of our log likelihood ratios. And then we find that their distributions of consecutive step lengths were long tailed, exhibiting patterns of short intensive searches. Coming to the discussions, we find that there is a preference for anthropogenically modified habitat classes, which is basically because of their dependence on provision food and garbage dumps. And this extended residence at localized sites, both roost sites and frequent movements to these refuge sites, could actually perpetuate heightened agonistic interactions in humans. And this non-Brownian movement strategy highlights the fact that even though they are a generalist species, they're considered to be a generalist species as being macaques, they also are using very specialized patches of refuge sites where they move continuously. And I would like to thank the following people.
In target project and the project biologist in 2019. Currently, she is working as a research biologist in the Dehradun project.
Thank you for the introduction. EDNA as a biodiversity monitoring tool. This study was conducted as a part of the project Development of Conservation Action Plan for Ganges River Dolphin. So before we move on, I would like to I would like to explain what eDNA is. So eDNA is the pool of DNA collected from an environmental sample. All animals naturally lose DNA in the environment in the form of hair, feces, skin, etc. So DNA is lost through all the processes required for life, like movement, reproduction, excretion, etc. This eDNA can be used to identify the species it comes from. So we have various techniques for identifying the species through eDNA. This approach is non-invasive in nature and is highly sensitive. It detects rare and invasive species. It helps in the identification of current biodiversity existing in the nature. So, I would like to stress on the point that there has been no previous studies on eDNA from India. So, we formulated our research objective from this. We wanted to evaluate the efficacy of eDNA as a tool for monitoring dolphin and aquatic biodiversity in Indian rivers. So for that, we chose Kulsi River as a study site. Kulsi River is one of the smallest tributaries of the Brahmaputra. It originates from the Khasi Hills in Meghalaya and meets the Brahmaputra at Nagarbera, Assam. It spans 72 kilometers in Assam and 12 kilometers in Meghalaya. It is home for a very small population of Ganges River dolphins. So the methodology which we adopted is, initially we sampled 25 liters of water from each sampling site. We filtered this water using vacuum filters through membrane filters, and then further we isolated DNA from these samples and proceeded with library preparation. Further, we adopted a metabarcoding approach as we wanted to focus on different taxa like fish, plankton, and vertebrates. For fishes, for all those taxa, we use different set of primers. For fishes, we
study are as follows. We found 6 families of fishes from our data, Cyprinidae being the highest one and Salmonidae being the lowest one. As we all know, Cyprinidae is the prey base for large fish species and also a food source for local communities. Sisoridae are indicators of water quality. We found 16 major families of vertebrates in our data, like Suidae, Delphidae, Hylobatidae, Equidae, Felidae, Canidae, Bovidae. And also, I would highlight that we also found Platanistidae, 4% of Platanistidae from our samples. We also found 22 major families of planktons from our data. They comprised of microzooplankton, rotifers, and diatoms. Microzooplankton regulate phytoplankton abundance and maintain balance in the ecosystem by consuming primary producers. Rotifers provide a critical link for transferring energy in the food web. Diatoms acts as a primary productivity and carbon cycling in aquatic ecosystems. So to conclude my study, eDNA has demonstrated its efficacy as a valuable tool for capturing both aquatic and terrestrial biodiversity within the river systems. Notably, we have found 4% of Platanistidae in our samples, which is the evidence for the presence of Ganges River dolphins in the Kulsi River. This detection aligns seamlessly with the field survey data, which says that 12 Ganges River dolphins are present in Kulsi River. We have found 22 planktonic families, which majority of them were 15 microns in size, less than 15 microns in size. This underscores the precision of eDNA, and we would like to stress on the point that there is a need to establish a comprehensive eDNA reference database or repositories so that there could be fine taxonomy classification of the species level classification. And eDNA has emerged as an invaluable tool for assessing biodiversity. I would like to acknowledge the following people. Thank you.
Now, before the next resolution by Star Rapporteur, who's talking on from pixels to conservation, automating reform and other marine conservation to artificial intelligence and machine learning. Sagar has completed master's in biology and currently pursuing PhD from he is working on a reform project.
Good morning, everyone. Thank you for the introduction. So today, I'll be talking about the topic from pixels to conservation, automating dugong and other marine megafaunal detections through artificial intelligence and machine learning. So when we started the Campa Dugong project, dugongs as a species has always been very challenging to detect. So it was very difficult with traditional methods like boat surveys. That is why we resolved to aerial monitoring using modern technologies like drones. So with the help of drones, we were able to detect not only dugongs, but a wide range of marine species like dolphins, sharks, turtles, etc. So when we would record these videos using drones, we would record it at a very high resolution. So the resolution that we recorded the videos were at 2.7K at 60 frames per second, which would lead to about 3.5 GB of video for a 5-minute recording with more than 20,000 frames. So in the survey efforts, we detected a lot of species which were of conservation importance. Like we detected endangered species like spotted eagle rays and zebra sharks, and vulnerable species like manta rays and dugongs, which are classified according to the IUCN Red List. So with this data set, we decided to make a tool that will help not only monitor but identify this species. So with that, we decided to take the approach of machine learning and artificial intelligence. So with the data set we decided to trim the data set. So for trimming, we used frame skip and dehashing methods. With frame skip, what we did was we took every 25th frame and discarded the other frames. And similarly, we use another data trimming technique, which is dehashing. So with this, we were left with a data set, from a total data set of 25,000 images, frames and 31 HD images. We selected 814 distinct frames
which was about 1,128 objects with the spread as shown in the image. Then we further used the supervisory tool to annotate the detections that we got. So we used bounding box as well as polygon annotations. With this, we uploaded the model in YOLO v5 to test the training and validation split with the 80-20 split. So for that, we got an overall accuracy of, we got an overall precision of 90% and a recall of 97% across 100 epochs. We also got a very high mean average precision level, which meant that the model that we created was able to detect this species. Then we did a test. So this was done using images and videos which the model had never seen. So we used about 6,000 frames to test the accuracy of the model. So in that, these are the results of the test data set. For species like dugongs and zebra sharks, we got an absolute value of 100% precision recall as well as F1 score. For dolphins, again, we got a very good value wherein the model was able to distinguish the species based on the test results. Moving to similar looking species like manta rays and spotted eagle rays, we got a very high value, and then again it was able to distinguish between similar looking species. Coming to turtles, the results that we got were not the best, wherein we got precision wherein the true positives were right, but when it came to accuracy, there were a lot of false positives. That is the reason why the recall and the F1 score was low, because the model was also detecting corals and other boulders as turtles. So this is the output of the tool that we have developed. As you can see, the animal here in frame dugong is not only being detected and identified, with each frame, the confidence interval is being shown as how confident the model is that this animal in the frame is a dugong. Similarly, like these are the results that we got. So like you can see, there are the waves and distortion cause a little bit this thing, but whenever it is clear to the human eye, even species like spotted eagle rays are actively being detected. So moving to the conclusions and way forward, this is the first study to detect and identify multiple species, marine species from Indian waters. It is an effective tool that can be used for monitoring and tracking of marine life by stakeholders like State Forest Department. It is a scalable model that can be trained to detect potential threats. So along with the species, it can also detect threats like fishing boats and nets, and more data of co-occurring species can be added. Right now in this, the model that we have developed, it's only based on the data we collected in Andamans, but similarly we are looking for having multiple data sets from Gulf of Kutch as well as Tamil Nadu. So with this, I'd like to thank the following people. Thank you.
We will talk about occupancy and acceleration of renewable degree larvae rush in the southern Gujarat. Suji has been working as a research biologist for the last 8 years in agriculture.
Good morning, everybody. I would like to present the topic about the occupancy and nest size selection of Nilgiri larpinthers in the southern Western Ghats. There are 4 species of larpinthers seen in the Western Ghats, and all, there are 4 species of mountain larpinthers seen in the southern Western Ghats, and all they are geographically separated with deep geographical gaps, and we can see the Nilgiri lapidars in between north of Palghat Gap to south of Chaliar River banks. The species is considered as an endangered species, and we can see the species in elevation above 1,400 meter in southern Western Ghats, and the native habitat of the species is shola grassland ecosystem over the tropical mountain evergreen forest, which is the native forest of the Nilgiris, and the entire area once covered with this native habitats, and all majority of the area has been converted for other land use purpose from the colonial period. And the remaining forest habitats are very fragmented into hundreds of small patches. In this scenario, the basic ecological study of an endangered and endemic species is very important to its future conservation. The first objective of the study is to identify the factors that influence the occupancy of the Nilgiri habitat in the southern Western Ghats, and the second one is to illustrate the factors that influence the nest site selection of Nilgiri habitat. The Nilgiri is the our study area, and methods for the occupancy. First, we identified the area above 1,400 meter and generated 4 square kilometer spatial grids. And all these grids are subdivided into four subgrids, and a 500 meter transit was laid within each subgrids. Then established 5 points within the interval of 100 meter, 100 meter, and 100 meter for detecting the bird species and sampling covariates and site covariates. Method for the nest site selection. Intensive nest search were conducted during the breeding season, and we quantified the nest and nest substrate characteristics, and compared the nest site and random site characteristic. We mounted the nest success for all the nests using camera traps. And coming to the result, we sampled 290 grids and species present in 51% of the grids, and high detectability recorded in the western part of the Nilgiris, which is the we can see the native forest are there in the western part mostly, and estimated occupancy was 0.56, and the detection probability was 0.48. Canopy cover and canopy cover, elevation, and herb cover are the mostly responding variables for the occupancy of the species, and we mounted 35 nest, and all are cup shaped. The mean diameter of and depth of the nest is 11.85 centimeter and 5.85 centimeter. The average nest height from the ground was 1.49 meter, and average nesting plant height was 2.13 meter. The clutch size was 1 to 4, and most of the nests had 2 eggs, and the nest success was only 43%. Most of the nests were placed on the shola edges, and 60% of them were on the shrubs. Nest dismantling was observed after the predation or failure of the nest. Canopy cover and litter cover positive responding for the key variables for the nest site selection comparing to the patch selection. And coming to the conclusion, area of occupancy of Nilgiri lavender is much higher than the current known area, and the species occupied mostly in the natural vegetation with minimal invasive species cover, and the Nilgiri lavenders preferred understory for nesting, and 43% of the nests were successful. And tree canopy cover and litter depth were influencing the nest site selection by the species. And thank you.
The next speaker is Ankit Mohan, and talk about situation level battery colors, food water in Bhavani.
Good morning, everyone. This presentation is a part of project we finished last year, smooth body water as an ecological indicator of riparian habitat ecosystem. So otters are the apex predator of riparian habitat ecosystem. They are keystone species as well, and they are excellent indicator of the health of rivers and streams. So the objectives for the presentation the project was to assess the riparian habitats' physical and chemical properties along the smooth corridor water distribution sites and to develop riparian habitat suitability map for conservation planning of the smooth corridor. So we chose Coimbatore Forest Division as our study area because of the heterogeneity. We had different type of forests there, and it was a mosaic of agriculture, settlement, and forest, and then most importantly, the different stream orders from first to fourth, which give us ideal condition to study orders. So we used three different methodologies. We collected data as direct and indirect evidence of waters by transacts and habitat variables using systematic plots. We use multivariate regression to see the impact of independent habitat variables on the dependent encounter rate. Then we used machine learning tool self-organizing map to see to categorize the African habitat, and then we used random forest to see the influential variable in the different clusters. Then the third methodology we used for habitat suitability, we used different models. We used our presence data which we collected over the time period, and we actually converted all the ground data into raster using rigging, and the same layers were used for the habitat suitability maps. So the regression results were almost similar for both the season with some changes. So for waters, barren land, emergent vegetation, high QBR score, which was basically riparian index, and stream temperature, islands, and degraded forest were very important wherever they avoided areas with high grass cover, shrub cover, and patches with high amount of chloride, ammonium, and nitrate. Similar in the wet season, the only difference in wet season was that they preferred forest plantation wherever they avoided moist deciduous forest and scrub forest. So this was the SOM result. The study area for both the season was divided into 6 different clusters, and each cluster was basically dictated by some influential variables. Like here you can see in the first cluster, the area of agriculture was high, regulator was low, riparian canopy cover was low. So similarly, in all the 6 clusters, it was different. The results are similar to the regression, but we got to see some spatial difference. Like we had both species data to compare smooth chloride and small chloride. So outside the protected area, all the clusters they didn't have any evidence of both the species. Smooth coated water was more confined to larger order stream, which was fourth, whereas small chloride they preferred second and third order streams. This was wet season, very similar. Then habitat suitability, dry season habitat suitability was very less, wherever it increased in wet season. And most of the habitat suitability it increased near to the confluent sites. So here we can see the extent of gain in wet season from dry season. Most of the sites were basically the confluent sites. So coming to the conclusion, so we could see the inverse impact of water pollutants on the distribution of waters, and least modified streams with high QBR, emergent shoreline vegetation, barren areas, and degraded forest were significantly important. They shifted, like preference shifted from dry deciduous to forest plantation in wet season. Wet season had more suitable areas and basically confluent areas. Area outside the protected area didn't have any evidence, and so the suitability was also very less. Lower order streams were more preferred by small chloride, and fourth order stream basically was preferred by smooth coated water. So the conservation strategy based on our field observation and like the results we got using our analytical procedures, that regular monitoring of water is very much required, basically in the areas with high industrial industries, especially in Metropolitan and Srimagiri regions. Feral dogs were in large numbers, so they need to be controlled. There was some hindrance in connectivity due to the lacking of the vegetation, native vegetation on some areas. So the monitoring of holds are very important. Here in background we can see one hold is destroyed by poachers, and fishing is also need to be controlled. And local tribers, right now the scenario is that most of the APWs they are more focused on the elephant monitoring, wherever the river areas they are not monitored well. So we basically suggested this to forest department that they can hire more anti-poaching watchers to monitor these areas, and long-term studies especially on the topic of water conflict. These are conferences we attended. I would like to acknowledge all these people. Thank you.
We have by talk about unlimited to Masters in Wildlife and Conservation Management from University of South Africa and currently working as a project biologist at WRI under the supervision of Dr. Sagar.
Thanks for the introduction. Good morning, everyone. While the Western Himalayan region stands as a place of profound ecological significance, but they are highly susceptible to the growing threats of anthropogenic pressures and effects of climate change. Although more than 500 studies on vegetation, on various aspects of vegetation, such as floristics, ethnobotany, community composition, assessing soil parameters and anthropogenic impacts, have already been conducted, but only a very small fraction of studies have attempted to document the effects of wild and canny factors together on vegetation. So in this study, we attempt to uncover the effects of biotechnical factors on vegetation in Chudhar Wildlife Sanctuary, which is a microcosm of larger Western Himalayan region. So in this study, our initial step involved the identification of different plant communities, and then we looked into what are the factors affecting vegetation attributes and diversity metrics. And we also looked at whether these patterns align with the intermediate disturbance hypothesis. So as you're all likely aware, this hypothesis suggests that diversity peaks at intermediate levels of disturbance, resulting in a unimodal shaped curve. We will see whether this theory holds true in the context of our study area, Chudhar Wildlife Sanctuary, where we conducted vegetation sampling using following standard sampling methods, and for disturbance data, we adapted the method followed by Kumar and Shahabuddin. We randomly laid 230 sites along the elevation gradient. Initially, we considered a total of 13 variables, 6 topographic, 6 biochromatic, and 1 anthropogenic. However, we had to remove some of these due to multicollinearity issue. For plant community classification, we used twin span analysis NAR, and to model the response of ecological parameters with predictive variables, we used generalized additive models that are capable of capturing non-linear trends in data. So these are the 7 distinct plant communities identified with our study area, as you can see in this graph along the elevation gradient. And this table presents the best models obtained for our ecological parameters and the deviance explained by them. And the highlighted variables are the significant variables, and the number of asterisks corresponds to the specific value, as you can see in this table. So now I will only talk about the significant variables. Along the elevation gradient, total visual area clearly formed a unimodal pattern, showing its peak value at mid elevation around 2,900 meters, where Lucretia mixed community dominated. While with solar irradiation and temperature seasonality, it formed multimodal patterns. And but we can see with solar irradiation, it formed highest peak towards the higher spectrum of solar irradiation, while with temperature seasonality, it formed highest peak within the minimum range of temperature seasonality. Tree density, initially, in tree density, we observe a gradual decline up to 2,400 meters, and thereafter it forms a prominent peak around 2,900 meters, where Lucretia mixed community dominates. With temperature seasonality, somewhere it forms a flat curve. It initially increases, but after the mid range of temperature seasonality, it starts decreasing. Tree species richness was found to be highest in the lower elevation range, as you can see, and then it abruptly declines up to the 2,400 meter, and thereafter it again rises, where we can see the dominance of Asia mixed community. With temperature seasonality, it shows somewhat a gradual decline with a peak after mid-range of temperature seasonality, where mixed forest community dominates. With disturbance, initially it rises at low disturbance levels, and then it again rises at higher disturbance levels. And we can see that at low disturbance level, the dominance of mixed corpus community, while at higher disturbance levels, you can see the dominance of Asia mixed community. Tree species diversity forms 3 peaks with long elevation gradient, one at the first at the lower level, second at the mid, and third at the higher level. And we can clearly see the dominance of different plant communities within these elevation ranges. vegetation, along with discoveries, shape the vegetation patterns in our study area. For total basal area, we clearly found a unimodal curve, while for other vegetation attributes and structural measures, we found multimodal patterns, and we did not find any strong evidence in support of integrated service hypothesis. These are my acknowledgements. Thanks.
The first question is for Sujin from Tamil Nadu. What's the explanation for nest dismantling behavior in Nilgiri lava crush?
Usually, from the nest, the bird feel any disturbance from the predators or any other nest failure, they will dismantle the nest.
Can you please explain why they would do that? They can just abandon the nest, right? Why would they dismantle the nest?
Sorry.
Yeah. Just wanted to know what the reason would be for dismantling the nest.
Actually, there are other studies needed for that behavior. Thank you.
Next question is for Akshay. Ayan, interesting study. Have you checked whether the change in connectivity as compared to model at all is due to forest cover or land cover change or refinement of the model?
This change in connectivity is due to the refinement in the model. It is because we used our model, machine learning model, we have the option to do the tuning, and we generated all our model from scratch, not using from an inbuilt software.
Thank you. Next question is for Sagar from this question is from Ayan. Interesting study in data accuracy. So they have two questions. Can the AI model detect multiple species in the same frame? Second question is, are you using any confidence threshold or score threshold while assigning species ranking?
Thank you for the question. So answering the first question, right now we don't have, we have just a couple of videos in which we were able to actually identify multiple species in the same, in the same video. So we had turtles and dugongs in like a couple of videos, and then the model was able to accurately identify both the species. And coming to the second question, yes, we are using a confidence threshold of more than 50% confidence, like 0.5 is the confidence value that we have kept. Anything where the model is more confident than 0.5, the model is able to detect the species. Thank you.
Next question is for Bhagna. Do you think whole metagenome analysis is more efficient in capturing species diversity and abundance than amplicon sequencing? And there is also a comment, DNA studies have been done in India, so looks like a lack of review in your study. From Umesh.
So, it was pertaining to DNA studies in river systems, Indian river systems, and I think amplicon sequencing is more potent than whole genome sequencing in estimating the diversity of the ecosystem.
Thank you. Yeah, you talk about the clean river system that EDN has not been done. So I would say it's a lack of review because the several studies done by the IT Kanpur, Delhi, and from Mohammed Central of Passport from Aurangabad. And in Delhi also, we have studied a couple of published studies. And when you talk about the empirical sequencing, so yeah, species capture and abundance, the diversity and abundance. For sure, I mean, when you are doing the whole the capture and the abundance is far better than the empirical sequencing because they, I mean, there are some factors in the individual DNA when you are doing the empirical sequencing. You are probably some data is like some species do not amplify the when you are doing the species empirical sequencing. Whole genome, metagenomics, I mean, it is two studies are there.
Next question for Surojit Mitra. Was density estimation done by using camera trap method substantiated by any other method to consolidate the data?
We showed camera trap distance sampling comparison with another camera trap method, which is random encounter model, but not any other method which does not use camera trap. So using the camera trap based two methods and taking YT as a model, we showed that there was no significant difference in density estimate. Thank you.
Next question is for Vidwan Borua. You discovered 8 new species. Please comment on the observed intra-specific variation you observed with their counterpart of the matching species. This question from Mukesh Thakur.
Hi sir, thank you for the question. You have are you asking this question for a specific group?
Irrespective of the eight new species discovered,
Yeah.
and we all know that this is I mean, very high endemic there.
Yes.
So, species that we discovered always tend to have a high divergence from the counterpart in the elsewhere and there. So, per se, even we have discovered these eight new species, so what is kind of the threshold that we got? Because these endemisms, we also in like in study rather than a lot of divergence from the counterparts. What is the threshold of the divergence you got?
So it varies actually for different group of taxons. For agamid lizards, in our paper, so in our study we found that these closely related to two species which I showed in the slide. These two species has high genetic divergence. In Andy2 it is more than 13%. But there are some other taxa which have very shallow divergence, or you can consider some brachypod frogs. So is it okay? Thank you.
Next question is from Akshay. Nice presentation. Can you please explain what approach was used to develop resistance surface since you have used habitat model as a conductance?
We didn't develop any resistant surface. Our input to the circuitscape model is the probability prediction map that is generated from machine learning algorithm.
Thank you. Next question is for Dr. Sunny. What is so special about dolphin oil that it should attract fishes? Why cannot we find an alternative? This question is from SS Raseli.
Actually, sir, I have mentioned in my presentation also that alternative oil bed has been already, it was introduced, and it was also practiced, and it was tested in the field. So the questionnaire surveys which was carried out among the fishermen have also said that there might be a very minute difference in the catches that has caught with alternate dolphin oil and dolphin oils. But the main case over here, which we are actually concerning, is the photograph which I have shared in my presentation. You might have observed the fishes, how it has been lured towards it when the fish bait is being used. So here we are actually, the main threat is to the population of the targeted fish, which is also declining because of this kind of practice. So my recommendation, our study recommends that there should be a total ban on oil-based fishing practices, not only dolphin oil, any kind of alternative oil should be banned from the country. Thank you.
Question is for Sagar. Next. Okay, so I'll shout. Next question. Mike. Next question is for Sagar. This is from Vishwa Singh. Type and make of drones used for the study. So there are three questions. First is type and make of the drones used for the study. Second, safe distance to be maintained to capture video without disturbing marine life. And third, AI model used during video capture.
Yeah, so the first question is, we used a micro drone, which had a weight of around 1.5 kg, less than 1.5 kg. It was a multi-rotor aircraft, which is very, so that was the drone that was used. Coming to the second question, so usually whenever we have, from the experience that we have with regards to disturbance of marine life, all this, since the drone that we used had zoom, we kept it at an elevation of more than 20 meters throughout the study, with a range of around, initially for detection, the drone would fly at 100 meters, and after the detection, we recorded videos with a range of elevation depending on 20. 20 was the least, and the highest that we had was around 50 meters to record that video with maximum zoom, that is around 4x zoom that we had with the drone. And third question was?
AI model that was used during video capture or post-analysis phase.
Yeah, so this was post-analysis phase. So this was basically based on frames. So the video is split into frames, and based on that, in each frame it is detected. So right now we have made a tool that actually can detect it live. Whenever we upload a video, like a media file, when the model is connected to the system, it will detect it in every frame. So based on that, it is a post-capture this thing. But then it can also be integrated with drones and other systems, live view as well. But we have not explored that possibility yet.
Thank you. Next question is for Tudala from Dr. Your study suggests ranging pattern for these macaques. Roosting is in WII, but feeding is outside. So why not analyze them separately?
Thank you for the question, sir. So actually, not roosting within WII, they are at different patches all across the study area within their range, and oftentimes their foraging sites and roosting sites coincide. So what I was hinting at was that there is a frequent movement from one of these patches to the other, which are dispersed within the home range, and that is why there is a strange distinction in the way they move around.
So we have 10 more minutes till we hit 10:30 tea time. So we can ask some questions. Okay. So there's just one question from Ayan for Bhavna. How far back in time you can go using the EDA technology?
eDNA is not stable. It only persists in the environment for weeks or max to maximum a month. So we can't trace it back to the history.
Thank you so much for all the presenters. It was quite a mixed bag, a lot of diversity of presentations as such. But I request our coach, SK Tavasira, to say a few words.
Thank you. First of all, I'd like to compliment all the 11 speakers who participated in the speech of I also appreciate the way they summarized very inspired by very knowledgeable session. Also like to compliment WI for coming up this kind of session. Actually, this is the first live session I had. I'm experiencing today this speed talk. Very, very informative and I'm very thankful to WI.
Thank you. Now, I will chair the whole session. I'm really happy to Master who really have chaired this and hope to
Thank you, Parag. I will begin by saying that what a delight it is to start the session at 9 o'clock with 11 beautiful speed talks. Interestingly, Rosita, there is a very good history behind these talks, and that is with the evolution of these annual seminar itself. When we started many, many years ago, these were only oral talks, and we used to decide whether these would be for 25 minutes or 15 minutes or whatever, whether these would be for completed projects or ongoing projects, because each one had a different flavor. And then as the number of presenters increased, we realized that not everybody could be accommodated for an oral talk. And then came the introduction of the poster session. So posters were introduced, and we thought that some of those will not be able to do oral, came invited to do posters, and then the posters increased in numbers, and so is the number of researchers in this institute. Then we introduced a digital poster, and we realized that there is a lot of money which is wasted in printing and doing that, so why not have digital? So these were the oral, then came the posters, and then came the speed talks. And interestingly, I would say that we run a master's course here, and the students do their presentation, and then they defend their presentation. And it was thought that some of the very good studies which come up through this MSc presentation would be brought in the larger ARS, because here the audience is different and the context is different. So the speed talks were introduced then, and the MSc students then were asked to make these speed talk presentations, which I would say were very well coordinated and received. And today we see that it's not just master students, it's open up to everyone. And I could see our new colleagues from the SECON also presenting that. So in terms of coverage, it has been super from study in the campus to ambulance in Northeast Asia and to many mammals and animals. Everybody has been covered. And interesting part was the use of technology. The use of AI and use of machine learning and use of environmental DNA. And there were quite some good questions from our colleagues from the other side who were asking about how the science of DNA is to move forward. So I would say it has been a very interesting session, and our 11 young researchers defended their bits, and I hope that in coming years the proportion of speed talks will increase. We can also think of adding more complexity to it. See, right now, everybody there was no restriction on number of slides. Somebody presented 10, somebody presented 15 or whatever. But next time around, if you bring the number down to 5, whatever you need to say it in 5 slides. You can also think of reducing 5 minutes to 3 minutes and add more. So the whole idea is to convince and to convey that why your research is important and what has been the finding. So many of us and our colleagues from the management side, they just want to pick up that what is that which is important, why this particular research is to be funded, because ultimately at the end of the day you need to decide on the funding part. So we need to look at the application part as well. So these are some of the ideas, but I thank you for inviting me for the session, and I think that these speed talks are going to add to the flavor of the AIS in years to come. Thank you.
Thank you so I now request our session co-chair, Dr. Parag Nigam, to kindly present a token of our gratitude to our session chairs and co-chairs. So first to Dr. Madhu, session chair. And now to our speaker. Thank you so much, sir. We will now end this first session. We will break for tea and also have the poster session outside. Let's meet back here after.
ये है 20 का ये आएगा। ये तो अबे ये है